Hey guys, welcome to Dr. Priyanka's Biology, where we discuss everything from biology to biotechnology. Today, let's get started with one important concept, that is regulation of gene expression. Now, why I'm saying this as important concept, that is because you take any board exams or any of your college exams, or you take your competitive exams, NEET or any of your competitive exams like CSIR or any other exam. This is very important concept. Now, if it has to come as a major question, it can come a question separately on regulation of gene expression, or if it comes as a 15 marks or some major question where you have to include about the operand concept too. So we'll be dealing with operon concept separately in a separate video so as to avoid confusions and becomes easy reference for you. So now let's get started with regulation of gene expression. So before we move on with the definition of gene expression, let us try to understand one simple concept behind it. Now we know that our cells, they have the same genetic makeup. Okay, irrespective of whether they are placed in your eyes or your stomach or any other part. Okay, they have the same genetic material, same DNA. Okay, the chromosome makeup is the same. But in spite of that, you see that certain organs, they are known to produce only one type of biomolecule or a product, whereas other organs, they produce some different type of product. So even the cells, they produce different type of product. How is that happening? Then secondly, if you see the amount of product required, for how much time it is required, everything is so very well planned. So when this planning goes haywire, or there is some problem in this planning or the sequence of the occurrence, then we say that there is something wrong with your body. That is how the infections or the diseases come into picture. So when we are talking about gene expression, okay, let's take example of the HCl that is being produced by the stomach cells, right? So now why this is required? So HCl is in turn going to activate the gastric enzymes which are required for the process of digestion. Now, just imagine the cells are the same, the genetic material is the same, okay? But still, why it is not produced in other organs, right? Why your pancreas are not producing HCl? Because it is not needed by the pancreas, right? Its function is something else. So we can say that in pancreas, your HCl, production of HCl is being suppressed or is being stopped, whereas its expression is being elevated or it is supported, okay, in the stomach cell. So we can clearly see that there is a difference. So that means there is some regulation, something is controlling, okay, some mechanism is controlling the entire process. Now this entire process, whatever is trying to regulate or put things in order, is nothing but regulation of gene expression. Okay, now if we see, for proper functioning of cells, all the necessary proteins must be synthesized at the right time, right? So the information needed to synthesize every protein needed by a cell is encoded by its DNA. But do you really think a cell requires all the proteins encoded by its DNA? Because see, the DNA is such a vast, one single gene, one single cell, it does not require all the products of this DNA. See, let's take one simple example. Now you are going for some wedding or some event where there is food has been arranged. So now suppose you are a vegetarian and the food served is mixed, both vegetarian and non-vegetarian. So you are not going to eat the non-vegetarian food, right? Or forget vegetarian, non-vegetarian. There will be some dishes which you do not like. So definitely you are not going to pick the dish which you do not like. So you will eat only what you like or what you want, right? So how is this thing happening? That is because you know what is to be done, right? So you will take only what is required. Same way it happens at the DNA level. So a cell does not require all the products of the DNA that a DNA is capable of producing because of its genes. Okay, same way it occurs in case of your cells too. So these cells, they are going to control the synthesis of proteins by turning on and off these switches. See? Suppose now you have a room, so you will put on the lights only when you want it and not when you do not want it, right? So you know you are going to control this process of lighting up the room by using the switchboard. You will turn on the button and off. Same way, the genes are being turned on and turned off only when it is required at specific time. So this entire process is called as gene expression. So now let us see what is the formal definition of this gene expression. So it's a mechanism that controls the time, location and the amount of product formed as a result of expression of a particular gene. So this is called as a gene regulation. So before we move on, there are certain things which I would like to bring it to your notice. So it may be a multicellular organism like a human being or a single cell organism like a bacteria. So this regulation of gene expression occurs to control which protein is synthesized when and in how much quantity. When should it be stopped? So this entire thing is so very well planned. See, whenever you are having food, okay, the entire machinery starts working. So when the food is not there, your HCl is not going to be produced. And even when your stomach is empty and HCl is getting produced, that means something is wrong. Your regulatory mechanism there's something wrong there. Okay, this is what occurs in your gene expression. So now this regulation of gene expression, what we call, okay, it is required. Now why this is required? First of all, it is required to save the space and the energy, because we know that turning on the genes, switching them off, all this requires the energy, right? So a cell is so very well designed that it knows that I have to save my energy to do the actual work rather than wasting my energy here and there. Then talking about the space, sorry, talking about the space. Now suppose if you see here, now consider this is the DNA that is present. Now we know it is wound, right? Now we do not require the entire DNA to be opened up. That is entire DNA. We don't want it to be unwound. Suppose now we required only this particular point here. Okay, suppose we required only a small location. Let's say, okay, we need only this much part of the DNA to be expressed, right? So we are not going to work on the entire length of the DNA, right? Rather what we will do, we will open up only this part of the DNA. Okay, this will be unwound so that the entire process occurs. The rest of the DNA it will remain the same, because just imagine if your entire length of the DNA has been opened, it will require more of the space to get occupied in that small cell. So this is what I meant to say, is to save the energy and the space, because all the products are not required, right? So this would require the entire DNA to be unwound if we want the entire all the products to be expressed, right? And it will take a lot of space. So this is not required. Then expressing only a few genes at a given time. So only when it is needed, that time only this gene should be activated, by only allowing that particular segment of DNA to undergo transcription or to be transcribed at a time. So this is going to save a lot of space of the cell. So this is one thing. Then next, Do you think this mechanism of regulation is the same for all the cells? So it would be different for a bacterial cell than a animal cell or a plant cell. Okay, this is because the prokaryotes like bacteria and eukaryotes like the animals and other things, they have different genetic compositions and the complexity level. Okay, so this is how it functions. So gene expression, if you see now, so it will be a process by which genetic information stored in the DNA is converted into the proteins, right? Because finally it will be the protein that is going to give the final expression. So we can say that expression of a gene, okay, how much the gene has been expressed can be directly correlated to the amount of protein that is being synthesized by the gene. So it is in direct proportion. So more of the gene expression, that means more of the protein has been produced. Okay. Now if you see regulation, okay, requires the regulatory genes. So they are of two types. One is the housekeeping genes and second one is the regulated genes. So what are these things? Okay, housekeeping genes. So in a hotel, we know there is something called as a housekeeping staff. So what is the role of the housekeeping staff? To maintain everything, the rooms, everything, right? So here they require, like they need 24/7, they need to be on duty, right? Because the guest can arrive at any time, right? So this, okay, housekeeping genes are expressed all the time. They are called as the constitutive genes because they are needed all the time because guests can come at any time, same way as that of a hotel. Okay, then the other genes, which are the regulated genes, they are non-constitutive, so they are not required at all the times. So their expression is such that it will be produced as and when they are needed. So only when they are needed, the switch will be turned on. So considering all these things, okay, gene regulation can take place at various steps of gene expression. Okay, so now this could be like, at what different levels it can occur, right? So first thing is it can occur at a replication level. So if at all there is any error in copying mechanism of this DNA, so it will lead to some altered expression, something else could be expressed. The next is transcriptional level. Okay, so what is this at transcriptional level? During transcription, so if there is any error in this polymerization, it will lead to altered expression of the gene, something else will be produced. Then next is post transcriptional level, so after transcription, so that is the modification time. So there may be it can occur during RNA splicing. Okay, so this will lead to further more changes. Then next is that it can occur at translational level. So during translation, so if there is any error in the attachment of the mRNA to the tRNA, then again it will lead to certain changes. Again, this gene regulation, it could be of two types. One is, it could be a positive one or negative one. Okay, so what is this positive and negative gene expression? So like see, we have a villain and a hero in a movie, right? So what is the role of a villain? The villain is going to suppress the good things. The goons are there, right? In a movie, they are fighting or they are creating problems. So their role is to create problems. So how they will create problems? Either by stopping the good work, right? Or by trying to control somebody who is working. So that means they are going to suppress or they are going to stop the good work. Sorry, whereas the hero is going to fight, right? So hero is going to do the good things. So he will try to bring up the good things. So same way, your positive gene regulation occurs with the help of something called as the activators, right? So these activators, they will keep the gene expression until that is need to be suppressed. So it will keep on going. They will say they are like cheerleaders, activators. So they will keep on telling that gene, okay, come on, go on, go on. So that is how it is going to work. So when the gene expression or the runner has reached the finish line of the game, they are going to stop the work. Whereas the negative gene regulation, when it occurs, like the expression of the gene is suppressed with the help of something called as a repressor protein. Okay, and it is induced only when it is needed. So like sometimes like you are going for some shopping or some fair, okay, when you were small, and your mother would be like, no, don't go there, don't touch there. So mother is kind of a repressor. So she will only allow you when you are needed. So this is how your positive gene expression and negative gene regulation occurs, right? So the inducer of a negatively inducer of a negatively regulated gene can be a metabolite, a hormone, or a substrate. Fine. So then we have the gene regulation in the prokaryotes. So this is a small topic here. Okay. So in prokaryotes, okay, like bacteria, cyanobacteria, or any other uh, prokaryotes. Their genetic makeup is how it is naked, lying in the cytoplasm. They do not have defined regions, right? So genetic material in most of the prokaryotes is composed of a single chromosome, right? So gene regulation here is the most extensively observed at the initiation of transcription. So the regulation of the non-constitutive genes is carried out with the help of regulatory proteins or the accessory proteins, okay, which affect the ability of RNA polymerase to bind to the transcription initiation site. So both the positive and negative regulation is observed in case of your prokaryotes. Okay. So now this entire study was done by Jacob and Monod in the year 1961, and they proposed the genetic material has a number of functional units called as the operons. Okay, now these operons, they are made up of certain genes which are regulated and operated simultaneously. So they will ask you to go also, and they will control the working also. So this way we have the operon model. So a operon is a functional unit constituting or consisting of the functional gene that are transcribed together to form a single mRNA that code for multiple proteins. So all the genes in operon are normally regulators, operators, promoter, and a structural gene. So we shall deal with this operon in our separate video. So till then, keep following us, like, subscribe and share our channel. So if you need any assistance in case of biology related topic, please get in contact with us. We will be always happy to help you. So till then, keep studying and keep smiling. All the best. Thank you.